Hello, everyone. I'm Colin O'Connor, Custom Content Editor at Genomeweb, and I'll be your moderator today. The title of today's virtual lunch and learn is Advancing ctDNA as an Early Endpoint Marker in Solid Tumors. The first 100 live attendees to log in today will be receiving a $35 DoorDash gift card that will be emailed out tomorrow afternoon to the email address used to register for today's event. Our presenters are Dan Norton, Director of Product Management at Personalis, and Diana Vega, Senior Director of Cancer Biomarker Development at Astrazeneca. You can type in a question at any time during today's event. You can do this through the Q&A panel, which appears on the right side of the presentation. And if you look to the bottom tray of your window, there are a series of widgets to enhance your event experience. And with that, I'll turn it over to Dan Norton.
Welcome everybody to today's Personalis sponsored webinar entitled Advancing ctDNA as an Early Endpoint Marker in Solid Tumors. Again, my name is Dan Norton, Director of Product Management at Personalis, and I'm very excited to be joined by the esteemed Diana Vega, again, Senior Director of Cancer Biomarker Development at Astrazeneca. Diana has an incredible amount of knowledge on this particular subject, so I'm really excited to hear about her insights and perspectives, and I hope you are as well. Before we get into the meat of today's discussion, I'd like to start with an introduction into Personalis and who we are. Personalis, we are leaders in advanced genomic testing. We're a public company that was founded in 2011 out of Stanford by some of the sequencing luminaries at the time. And since then, we have sequenced over 500,000 human samples, ranging from whole genomes to transcriptomes to exomes, targeted panels, plasma, etc. We are headquartered in Fremont, California, just south of San Francisco, in a 100,000 square foot lab and office space, and have worked with, I'm sure many of you on listening in. And if we haven't worked together yet, certainly look forward to doing that. We are prolific in publishing data from our advanced solutions with over 100 publications in peer-reviewed journals and posters. And all of that data is produced from our CLIA CAP in New York State credentialed lab, again, headquartered in Fremont, California. With regards to some of our capabilities, we offer advanced blood and tissue products in both the clinical and biopharma segments. We'll get into that more in just a moment. We have a very highly experienced regulatory and quality team that has developed a very robust infrastructure to support clinical trial assays and companion diagnostics. And it's that same team that is experienced also with regulatory body interactions and associated filings. To support the current trials that we are involved with and future trials, we also have a dedicated clinical trial operations team to manage trial logistics with global reach. So we have the solutions to help support you and your efforts to develop innovative therapies. Now, just taking a brief look at our product portfolio, we offer a variety of solutions for both biopharma and clinical applications. In blue is for biopharma, which we offer both tissue and blood-based products. On the tissue side, we offer our flagship immunogenomics profiling assay called AminoID Next, which is an exome and transcriptome-based solution that can be used for advanced tumor profiling, as well as it's being heavily used as the front-end sequencing for personalized cancer therapies. An example of that is public is with Moderna. And then on the blood side, our flagship MRD and monitoring assay is called Next Personal. And there are various flavors of that, from RUO to IUO, depending on the need. These products are also offered to patients through their doctors. The doctors order NextDx for therapy selection in the late stage setting, which is Medicare covered, and also Next Personal DX for early recurrence detection, and is also Medicare covered in breast cancer. Now, I wanted to talk a little bit more about Next Personal, and I wanted to start here Next Personal, several years ago, many years ago now, was inspired by the need for more sensitivity. So focusing on the left-hand side, we often see Kaplan-Meier plots that look like this, where a sample is taken in the perioperative setting. And say in this example, if it's post-surgery, and we see a ctDNA negative line that looks like this, where there's lots of relapses that happen over time. And so the question is can we more accurately classify these patients? Are those patients that were negative after surgery in this example really negative? Or were the assays used at the time not sensitive to detect the ctDNA at that time point? Ideally, what you'd like to see in the negative line is a very flat, straight line. And you're going to see that later in this presentation with use of Next Personal. The other question that we want to answer is, can we detect ctDNA sooner than a clinical relapse through monitoring? And it's likely that you've seen patient vignettes that look like this, where after a surgery, in this example, patients become ctDNA negative, only to emerge later in time as positive. So were those patients actually negative when they were testing negative, or were the assay not sensitive enough to detect them? So how does Next Personal work? I'll orient you to the top. First, Next Personal is the pioneer and is the current leader in ultrasensitive ctDNA detection. And we define the ultrasensitive space or range as less than 100 parts per million. The way that Next Personal quantifies ctDNA is using parts per million, which is defined as the number of tumor molecules sequenced per 1 million total molecules sequenced. So here's how it works. We analyze each patient's solid tumor using whole genome sequencing up front, not whole exome. With this much larger, broader genomic space, it allows us to identify up to 1,800 unique DNA markers, somatic markers, that are present in the tumor. We then go on to make a personalized panel for each individual patient. And with those up to 1,800 markers, sequence the plasma, which allows us, with these 1,800 markers, to detect as low as 1 part per million, or 10 to the negative 6. Now, the 1,800 markers, one of the key benefits of that, as I've just mentioned, is the exquisite sensitivity. But to orient you to the bottom left-hand of the slide, that it also delivers consistent LODs in the 1 to 3 part per million range, regardless of cancer type. So the data that you're looking at the bottom left is real-world data from Next Personal DX, again, the assay that is ordered by physicians for their patients. And we plotted the LODs across a variety of different cancer types, colorectal, breast, et cetera. And what is striking about this is just the really, really tight band of LODs, regardless, again, of the tumor type. In other approaches, such as phased variants or structural variants, often what you may see, or at least what has been published, is a broad range of LODs for a particular patient across multiple time points or in a cohort itself. So here we're just juxtaposing that to our very, very flat and tight bands. So when we say 1 part per million, I'm not saying down to, we are always in that 1 to 3 part per million range. The 1,800 markers also delivers 1 to 3 part per million range, regardless of the input amount, which is the center or the middle chart. So even if we decrease the amount of plasma cfDNA into the assay, there's only a very small marginal impact on the LOD. Finally, how does that translate to detection in cancer itself or cancer patients itself? In a large, and I would actually say the world's largest non-small cell lung cancer consortium called TRACERx, led by Dr. Charlie Swanton, there were 3 different studies that were performed not on the same patients, but within the same patient population, using 3 different MRD assays, which is our closest way to compare the detection rate preoperatively. In 2017, there was a 16 target assay that was used. 2023, there was a 200 target assay that was used. And then recently this year, NeXT Personal was used. And you can see that across all stages 1 through 3, and combined, there was a 2 to 4x increase in the preoperative detection rate. So again, this is where the ultrasensitivity starts becoming very important. Just 2 slides left before I turn it over to Diana. I want to really, I think this is a really important point, is that NeXT Personal is also analytically validated and published in a peer-reviewed journal. There's an image of that journal there on the left-hand side. We published this in 2024. And some of the key takeaways are, number 1 is that this is one of the few ctDNA MRD assay validations that have been published in a peer-reviewed journal. So I would encourage you, as you're looking through or looking at different assays, to ask, is the assay validation completed, and if so, is it in a journal? The analytical validation supports the use of NeXT Personal both as a clinical trial assay and as an LDT. The validated performance, as described in the paper, is greater than 99% analytical, 99.9% analytical specificity with an LOD 50 of 1.67 ppm and LOD 95 at 3.3 ppm. This is all done in concordance with CAP FDA and recent blood pack guidance. Again, we're very transparent about the assay, about its reproducibility, the various measurements that we took from LOB, detection threshold, LOQ, et cetera, and encourage you to take a look at this paper. Lastly, I also want to point out that NeXT Personal is the most clinically validated whole genome powered ultrasensitive MRD approach today. This is just a sampling of different studies that we have performed with some of the top KOLs in the world, from Charles Swanton in non-small cell lung cancer to Nicholas Turner in breast cancer, et cetera. Many of these studies have been published in Cell, Nature Medicine, Annals of Oncology, et cetera, et cetera. And one of the really exciting things is that there is a deep pipeline of studies across various cancer types that are in progress now. So you'll be hearing a lot more from Personalis as we continue to build evidence for the use of ctDNA across a variety of use cases and applications, both in interventional studies and retrospective as well. So with that, I would like to turn it over to Diana to talk more about ctDNA as an early endpoint in solid tumors. Diana.
Thank you, Dan, for this opportunity to speak about something that I'm very passionate about, which is ctDNA as an early endpoint. And it all starts with the patient being the North Star. The motivation for the use of ctDNA as an endpoint, particularly in early stage tumors, is because there's a lack of early and biological meaningful endpoints that reliably predict a long-term clinical benefit. So we have currently various ways to measure whether a patient is responding to a therapy. Some of those are radiographical in nature, but there are some concerns with the limited sensitivity, particularly in the post-surgical window or where there's a low tumor burden of the disease for the radiographical response to be captured accurately. There are some other pathological-driven response metrics, such as PCR and MPR, but it may not work across all tumor types and therapies. Also, there are biomarker endpoints such as PD-L1 or Ki-67, PSA, whatnot, that offer some predictive power, but sometimes this is not as accurate as we would like it to be, as sensitive or specific. So the goal is that knowing that ctDNA is a good proxy for tumor burden, capturing both activity and tumor size or volume, that ctDNA can help compress trial times, particularly in the early stage disease setting, where we have long periods of time to wait until an overall survival metric or an event-free survival metric. And also, ctDNA can more accurately predict risk and risk of reoccurrence, for example. So there's an urgency to accelerate access to potentially life-saving therapies to patients that have significant unmet needs. And we're going to be talking about that a little bit more because that drives where is it that we can start thinking of this and pitching as a first place to start looking at ctDNA as an early endpoint. And also an opportunity to expedite drug development and lower trial costs. And this is also best for the patient because that means patients will have options, and they'll have options quick enough without having to wait for an overall survival of 5 to 10 years. So let's start. This is something that I'm very passionate about, and it's to start from the start, definitions. How do we define ctDNA as an early endpoint? And we need to understand this because this will help us align on a fit-for-purpose approach where we look at ctDNA. We know ctDNA is quite dynamic. We see it and we call it detected. We can also quantify its value. And depending on the assay's limit of detection, you can see the actual ctDNA values decrease or become non-detected if the patient has responded to treatment. At this point, this patient's ctDNA has cleared. It went from detected to non-detected. If the patient was always non-detected, because that's sometimes the case, we don't usually use the word clear. It's just non-detected or low VAF or low ctDNA values. But then eventually we see what we're coining as molecular relapse. And this is where ctDNA emerges from it being non-detected at a first time point or a prior time point to then becoming detected at a later time point. So you could potentially be looking at ctDNA as an endpoint by looking, for example, at the sustained clearance of ctDNA, meaning a patient had ctDNA before and after the therapy, it cleared and it sustained clearance at a particular amount of time. That could be considered an endpoint. Or we could be looking at ctDNA reemergence, so going from a non-detected to a detected time point, and that could also be known as an endpoint. And that's very important because, as I said before, it's important for us to understand the application, the biology, what questions are we trying to answer to best understand what definition of endpoint would best serve the situation. Another thing that we need to ask ourselves is, again, going back to the biology and the question, where would ctDNA be the best tool as an early endpoint? And here we need to talk about unmet needs. Unmet needs in early stage cancers point at indications that are likely to benefit from ctDNA as an early endpoint. But again, there's different ways to look at unmet need. And here there is data from SEER that has been evaluated. And as you can see, we have different indications on the left. We put a line through it because you can see that there's some indications that have longest time to death. And you can see that their recurrence times change across the different indications. So you can see longer recurrence times in the purple and longer time to death in the dark brown in the top indications compared to the bottom indications. So one can say that those indications with the longest recurrence and the longest time to death, they may benefit from ctDNA to shorten trial duration because this can support identification of events ahead of only looking at overall survival, which may take a long time to reach. But then you can also look at it as an unmet need for those patients with rapidly progressing or very aggressive disease, which have the shortest recurrence and the shortest time to death below that line. These are highly aggressive cancers where imaging may not be sufficient, or it may be that we can know earlier on, whether the patient is responding to therapy or not to be able to manage the disease accordingly. And why do we insist in ctDNA becoming an early endpoint? As I mentioned before, there's radiographical based endpoints, some other pathological based endpoints, but why ctDNA? So what are the reasons to believe? More and more, there's been data and evidence that has accumulated over time to show that ctDNA can be a more accurate predictor of recurrence risk. that it outperforms some traditional markers of recurrent risk like PCR in the case of the PREDICT study that looked at TNBC, as you can see on the left panel there, that it predicts relapse with larger lead times versus clinical relapse. As you can see from the Garcia Maria's paper on the panel on the right, looking particularly at how long ahead of clinical relapse did a molecular relapse occur. And this could be several years, 2.6 years in this case, to relapse by imaging. So there's an opportunity to increase the NPV, the negative predictive value, and the specificity using ctDNA to non-relapse patients, and thus identify patients who truly need therapies or could then not have as much therapy and spare that to the patient. So ctDNA, very exciting, and we believe that it will make a difference. And I also wanted to explain a little bit about the FDA's view on surrogate endpoints to make sure that we're level setting that information across everybody who's speaking about this topic. So the FDA looks at validated surrogate endpoints that lead to traditional full approvals by looking at both a trial level association and individual level association. There's cases where the data available to prove the validity of a surrogate endpoint is not as large or as much as we would like. And in that case, the surrogate then becomes a surrogate endpoint reasonable likely to predict clinical benefit. And that can lead to an accelerated approval, but that would still, like any other accelerated approval, require confirmatory trial to reach the traditional or full approval. And I just want to make an asterisk there that MRD, minimal residual disease in multiple myeloma, as the surrogate endpoint reasonable likely to predict clinical benefit, as you can see on the right. This trial level association, and the difference between trial level and individual level, is that individual level association looks at the strength of the association between the candidate surrogate endpoint, in this case ctDNA or MRD, and the true clinical endpoint. Basically, it's independent of treatment, and as such, it's called prognostic. But the trial level association looks at the delta, so the difference between curves. It can only be done in randomized controlled trials. It looks at the strength of the delta, the association between the treatment effect on the surrogate and the treatment effect on the true endpoint. So basically, it's saying, is the delta that I see in that early endpoint similar or the same as what I would see if I waited for the traditional or true endpoint? And this is predictive because it's very dependent on this delta between the treatment arm and the placebo or comparative arm. And obviously, depending on the strength of the evidence, the FDA can then judge whether this would be a validated surrogate endpoint or a surrogate endpoint reasonable likely to predict clinical benefit. So now that we've covered all that information, that's important to set the right context to potentially look at proposals. And here we wanted to ask some questions, and we wanted to ensure that we are looking at it from different perspectives. But what if we potentially pitch ctDNA as an early endpoint in early stage lung? We need to look at things like unmet need. What are the potential applications? What is the data availability to then run a meta-analysis, which is required by the FDA to be able to determine whether that could be a reasonable endpoint, early endpoint in these settings. So let's walk through this. Let's walk through the example of lung cancer. Let's say a proposal, a strawman proposal on what would it look like, or what would it take for ctDNA to be considered an early endpoint in early stage lung. The potential applications in early stage lung could be the following. ctDNA could be looked as an early readout for adjuvant immunotherapy and their targeted therapy benefit. So we could be looking both at a reduction in ctDNA or a reemergence in ctDNA to assess the efficacy of the adjuvant component of, in this particular case, early stage lung cancer. The application could also be an early indicator of neoadjuvant response before surgery. So in this space, we would expect to see a reduction in ctDNA caused by the neoadjuvant therapy. We could also be looking at post-surgically, whenever we're talking about residual disease and the post-curative intent, whether MRD can help guide escalations or de-escalation. And in this particular case, we could be looking at ctDNA reemergence after surgery, so that would be at the post-surgical landmark time point, or it could be during adjuvant therapy or in the surveillance setting after adjuvant therapy. So that's the reason why it's important to understand the dynamics and how those could be used differently as an endpoint. The unmet need in lung cancer, we currently have certain endpoints like PCR or NPR that are useful, but they're not necessarily universally validated. So there may be an opportunity for ctDNA to bring in additional value there. What about the heterogeneity and responses seen, particularly to immunotherapy? And can we trust radiological responses? Is there any way that ctDNA can help adjudicate and issue the progression there? And what about early decision-making tools in this particular setting, as these are limited? Perhaps ctDNA can bring value there and meet that unmet need. Lastly, let's talk about data availability, because we need randomized controlled trials that have collected ctDNA prospectively in order to run this. We see the landscape. We see there's many different data sets with strong correlative data where this could potentially be utilized and observed as a meta-analysis. So there is some data available to be observing or analyzing ctDNA as an early endpoint there. But we also need to ensure that all of this data can be analyzed together. So there needs to be an evaluation of how the cross-trial collection of time points can be standardized, whether it's standardized on these ones that have read out or any that will read out in the future. And that's something that is very important to make sure that we obtain the best pool data, but also the best meta-analysis data. Mm-hmm. And Dan, I don't know if you want to comment about this slide.
Yeah, sure. Actually, before we move to here, I'd like to just go back one. I wanted to ask you a question, Diana, about reduction, in particular reduction, or emergence even. When you think about reduction, do you think about it as a full clearance in the early stage? Or do you think about it as a reduction is like there's a certain threshold, like reduction of ctDNA from one time point to another of, say, greater than 30%? How do you think about thresholds, or are those things that need to be established?
That's a great question. And I think that that's particularly relevant in the space of ultrasensitive tests, because we are starting to see, and again, I want to point at the fantastic work that Forensic Cancer Research has done through the CT Monitor project. In the CT Monitor project, they looked at advanced lung cancer, and some of the work that was done there defined that in the advanced lung cancer space treated with immunotherapies, something as limited as a 50% reduction in ctDNA was associated with OS and PFS. And that's in the advanced setting. Obviously, if you have earlier disease, you would expect to have less ctDNA. Would it be easier to clear the ctDNA, particularly at lower limit of detections if the levels of ctDNA to start with are quite low? Perhaps. I think that's definitely a question that needs to be further evaluated, particularly in the context of early lung disease. But in the space of advanced lung disease, the work from CT Monitor is already showing that there's greater association with long-term survival, the more strict your definition of reduction is. So let's say 90% over 50% and clearance. The only caveat I'll say that is that clearance or complete reduction detected to non-detected is very dependent on the assay's LOD. So it's tied to the assay's LOD.
So is this then where an ultrasensitive approach becomes very useful because of, there's this wide range of LODs to really, elucidate this the small fraction of ctDNA in some of these patients.
So I think that the ultrasensitivity of the assays definitely enables us to better understand because of the greater dynamic range. And that greater dynamic range will play to our advantage whenever we're trying to understand exactly what is going on with the patient.
Right. Okay. Thank you. Well, I want to move to the next slide. So related, I want to just provide you with an example. I mentioned the TRACERx study, which was early in the presentation, which is a study in early stage non-small cell lung cancer led by Charlie Swanton and James Black, et cetera. This is data from Cell that was just published a couple of weeks ago, where NeXT Personal was used in both the adenocarcinoma and squamous or non-adenocarcinoma non-small cell lung cancer. Again, this is stages 1 through 3. And the point of showing the swimmer plots on the left-hand side, whether it's adeno or non-adeno, is not to dive into any individual patient. Rather, it is to show that, for example, on the relapsing side, which is the left side of the panes, that those that relapsed, virtually all of those patients were detected as ctDNA positive with NeXT Personal. But just as importantly, on the non-relapsing patients on the right side of those two planes, you see a lot of open circles, which is ctDNA that was not detected. And for those that ctDNA was not detected or had converted from positive to negative, because many of them were on adjuvant chemotherapy, this is exactly the kind of data that you want to see that helps support ctDNA's use as an early endpoint marker. And then looking on the right-hand side is a Kaplan-Meier plot from the same study that's looking at longitudinal ctDNA detection, and this is any time post-operation. And particularly, I want to point out the persistently negative line. So these are patients that were ctDNA negative post-op and on. Again, here's where I think is a very powerful example of, if you are thinking about de-escalating therapy or using ctDNA as this early marker, that trusting the negative and trusting the positive is really, really important. And so this is data that I think supports that notion. And so, Diana, I wanted just to ask you, when you look at data like this, what sticks out to you in the context of ctDNA, or the context of an early endpoint?
Well, the first thing that I can say about this data, just looking at those swimmer plots, I just stare and I say, wow, that's a lot of data points, and it's very rich data, which is fantastic. But as you say, I think whenever I personally look at swimmer's plots, I take this very personal because every single line is a patient. And here you have patients that are recurring very quickly. So those are the patients that have very tiny little lines, and you can see that they're pretty much positive for ctDNA throughout. So if you knew that they were positive throughout, then could you act quickly to then do something? And that's the power that ctDNA has. And then, like you were saying, in those that are non-relapsing, you do see that there's 5, 7, 10 time points for which these patients are negative. And they're not relapsing, at least until, whenever this follow-up was there. And I see already that there's potentially some positives at the very end of the follow-up period. So you actually wonder what would happen to them in the future and whether they will relapse. So I think that that's something that I look at. And obviously, whenever you're looking at Kaplan-Meier curves, you always wonder whether you could do better, whether you can get that persistently negative curve closer to 100% all throughout, so that you can better disambiguate those that are in the middle categories, and you have the persistently positive that are not doing very well. But then you also have those that convert positive or convert negative, and those you can start making some difference in their outcomes. I think that's where my mind goes whenever I see these data.
Mhm. Thank you. Thank you for the perspective. Okay, let's talk about breast cancer.
Yeah, it's a topic or an indication close to my heart. And Dan, I have to say, this hits home. I have a very good friend who was recently diagnosed with HR positive, HER2 negative breast cancer. She's actually having her first infusion tomorrow. So this is something that definitely of interest for so many reasons. But again, putting our hat on of like, what would happen if we wanted to get ctDNA to be an early endpoint in early stage breast cancer? What do we know about the unmet needs? We talked about the applications very clearly before and during the lung proposal. But here in breast, like, what are some of the unmet needs, particularly in the adjuvant setting? Because we're thinking early breast cancer, these are patients who do quite well for a long time, but the chances of them relapsing is quite high as well. So if we can basically shorten that by predicting using ctDNA what their natural history of disease could be, perhaps we could be generating better trials that are more expedited, but also ways in that we can be anticipating their recurrence in a chance that gives patients an opportunity to do well previous to an actual clinical relapse. So let's go from top to bottom. Subtype HR positive, HER2 negative, long history of disease with late recurrences, and also very variable. The recurrences can happen 9 months after surgery or 9 years after surgery. So is there a way for ctDNA to anticipate that the recurrences, whether they happen 9 months in or 9 years out? There's also a need for early markers of endocrine therapy effectiveness, and disease-free survival events take a long time to accrue. So this leads to long and very expensive trials. So I think there's an opportunity for ctDNA definitely to make some inroads here and be quite valuable. For HER2 positives, the unmet needs, similar to this, there's standard adjuvant escalation pathways now with more choices for patients, which is always good. And there's need for markers to avoid, obviously, over-treating patients, particularly who probably didn't need that extra adjuvant therapy to do well. And in the triple negative, obviously, it's a more aggressive situation, a more aggressive subtype. There's a high relapse risk even after chemo is given to these patients, those that are unable to receive it. No reliable biomarkers, again, to guide escalation or de-escalation in this particular setting where we have options. And again, in all cases, overall survival and disease-free survival or event-free survival require long follow-up. So ctDNA can certainly provide value there. And there are data sets available that could potentially be utilized for these meta-analyses to better understand the biology, obviously, but understand, as we were saying before, the delta between arms and whether the delta between arms in the early endpoint equal or similar to the delta in the actual true endpoint. So I think there's lots of great opportunities in early breast cancer as well.
Excellent. Yeah, and also in the neoadjuvant setting, we didn't cover neoadjuvant so much here, but I think that's also an area of opportunity too.
Well, and we talked about it in lung. We didn't talk about it here, but I think both of those apply because now we're moving to a setting where you have perioperative regimens. So it's a way of understanding the neoadjuvant, whether they're responding to that neoadjuvant regimen. After that, comparing it to PCR, and in some cases, ctDNA has been shown to be even more predictive of long-term outcomes than PCR. Can we have these patients go through an observational arm and not have additional treatment if they don't need it? Can we escalate those that need it most? I think ctDNA is quite valuable to be able to answer all these questions.
Right. Yeah. And you showed some of that data earlier in the presentation, the PREDICT data in particular, that showed this really large delta in hazard ratios between PCR only and ctDNA only, which is recapitulated in other studies, particularly in triple negative, like the Scandari study out of Francois Clement Bedard's group, I Spy, et cetera, et cetera. So yeah, certainly I think there's a lot of opportunity for that biomarker in the neoadjuvant setting for early stage breast. On the next slide, I'd like to show also some next personal data, particularly in the adjuvant setting, which was performed by Nicholas Turner's group at Royal Marsden. And this study in particular was authored by Garcia Morales. On the left-hand side, again, we see patients that were positive go on to relapse and patients that were negative not relapse. Again, so it's this theme of the ultrasensitive test that can really help accurately classify risk of relapse. What's interesting about this data is that I'm focusing on the left-hand side, but now in pane B, and then I'll talk about pane C next. The baseline detections were much higher in terms of the quantitative amount. Notably that many of them were much higher outside of the ultrasensitive range, but there is a good amount still within the ultrasensitive range. But the story switches once these patients are being monitored after surgery or on therapy, where the amount of ctDNA is being reduced dramatically down into the ultrasensitive range. So again, just demonstrating this need for ultrasensitive testing to pull out true positives. In any case, moving to the upper right, I was talking about Kaplan-Meier, a Kaplan-Meier plot at the beginning of today's talk, where one thing that we're trying to solve for is finding the true negatives and trying to avoid false negatives. And so these two Kaplan-Meier plots you see in the upper right-hand corner, RFS and CSS, which is cancer-specific survival, it's like OS. It's exactly what you want to see, these perfectly straight negative lines. And again, this is underscoring that need for ultrasensitivity to accurately classify the patients. And not only that, but as we go to the bottom of the slide, the lead times to recurrence, there is significant lead time to clinical recurrence in those that did recur, which I think to your point earlier, Diana, is one of the key goals here is to contract these trials as much as possible to get drugs faster to these patients. And then lastly, in pane D on the bottom right is this, from the same paper, is a relationship between the quantitative ppm value at the first post-operative detection and the remaining survival time. So on the y-axis there is the amount of ctDNA, i.e. the quantified amount, and then on the x-axis is the time to survival. And what this is showing us is that first post-operative positive detection, that those that were really high in ctDNA had a much shorter time for survival than those that were much lower. And so when we're thinking about risk, and we're thinking about de-escalation or escalation or whatever we're trying to do in the study, it's the amount of ctDNA that helps dictate or seems to dictate these lead times to recurrence. So again, taking a step back, Diane, I wanted to ask you what your thoughts are, like when you again see data like this, in this case is in breast.
Yeah, when I saw this on the paper published by Isaac Garcia Murillas, the first figure that caught my eye was D on the bottom right there, because it was very clear that the amount of ctDNA that is measured, in this case in ppm, when it's first detected post-surgically, is directly tied to the remaining survival time of that patient. So if we can't see the very low, like if we can't detect ctDNA at very low levels, then we are already identifying the patient which has ctDNA at high levels that we know is not going to have a long remaining survival time. So if we capture those patients when the ctDNA level is quite low, we have the best the ability to identify patients early and provide a potential for cure potentially in that case. So act quickly before it's too late. So I personally like that. And it was very interesting to see the y-axis because the ppms are really low, and you can actually see how the lower they are. So the bottom of, like the dots at the bottom of the plot tend to be more on the right side, which means they have longer survival times than those that are detected when the ctDNA is already high in circulation.
Right. Yeah, that's a great point. Okay. I'm going to move on. I think this is our last slide. I want to turn it back over to you, Diana.
So, as I said before, I'm very passionate about this. We've been talking about this for a long time as a field. I think there is a lot of momentum, but what would it take? What would it take? What are the steps? What are potential roadmap steps that we can take in order to select the space where we're going to work on this? So I've put 5 points down to make sure that people have an idea or a sense of what it would take to get ctDNA in front of the FDA to be asking ourselves, can this be an early endpoint? So my number one, always patient-driven, understanding the unmet need. Where can ctDNA bring the most value? Understand what's the greatest unmet need and focus there. Number two is prospectively understand where the data lies, because we obviously need data that is prospectively collected from randomized controlled trials. This is stuff that you can't really make up in a day. You need, in this particular case of early stage disease, long follow-ups to be able to understand what the relationship between the early endpoint and the true endpoint, which most of the time is overall survival, and it can take decades sometimes. So understanding where that data is, and if we need to generate that data, is understanding who can generate that data and how can we collect the data in a way that can be analyzed in the future. And I'll talk a little bit more about that, but collection time points is key. Those are things that you can't go back and say, oh, geez, I wish I would collected at this time point so that we can analyze what the association of ctDNA as an early endpoint is there. And so here I've talked about the support, the need to show support of the use of ctDNA as endpoint reasonably likely to predict long-term outcomes. So that's why it's important to understand where do we have the bolus of data necessary to start running some of these meta-analyses. We need analytical partners who can look at this evaluation, this meta-analysis of RCTs. If we need to bring randomized controlled trials together, that means multiple sponsors collaborating, donating their data. Who is that neutral third-party broker that can analyze these meta-analyses and demonstrate both the individual and trial-level associations with survival outcomes? I know how prickly it is to be able to share data in a way that is open and transparent, and it has been done in the past. Forensic Cancer Research has done it successfully in the advanced setting, so it can be done. Multiple myeloma has done it, and where does the FDA stand there to motivate the sponsors in order to share the data? Number 4, alignment on definitions, time points, criteria. All of this is so important, and it is up to us, the field, to get together and put some data sets together to have these data-driven decisions as to what should constitute the definition of my endpoint. Am I looking at a sustained clearance? Does that mean a single time point, 2 time points, 3 time points? Does that mean a year after surgery, 2 years, 3 years after surgery? How do we determine what long-term means and the association with an OS benefit? What about ctDNA reemergence, and how can we test that to predict long-term OS outcomes? In this case, the reemergence would be associated with a poor outcome. As I said before, collection time points so important, as well as assay depth, which is something that in the case for multiple myeloma, they were not even looking at the same thing. Some tests were looking at flow-based tests, the other ones were NGS-based tests, but they could all reach down to a level of sensitivity that was common. In this case, it was 10 to the -5. So can we do something like that and draw a minimum threshold? And fifth and last, we need to understand the biology. What is it that we are looking for? Lung is not breast, and even within breast, there are different subtypes that are not all the same. So understand a little bit more about how does this tumor shed? What is the rate of false negatives? How can we maximize this? Obviously, using an ultrasensitive test can help reduce that number of false negatives and better understand that for the particular application of choice. So these are my 5 points. Hopefully these resonated with the audience, but always happy to talk about this and better understand ways in which we can move forward so that we can create an impact for the patient.
Awesome. This was, I love the way that you just walked through this, Diana. This was so insightful. And as we wrap up, I just wanted to say thank you for lending your knowledge and your perspective and your insights into a really, what I think is a very complex topic, but one that is very important to help get these drugs that sometimes take a long time to develop to patients that need them as quickly as possible. And I am a firm believer, and I mean, I am biased, of course, but I'm a firm believer that ctDNA does have the opportunity and potential to transform, the way that clinical trials are done. And to your point, the more data that we have and the more collaborative that different sponsors can be with each other, like we saw in multiple myeloma, the faster we can, use ctDNA and bring these drugs to market faster. So again, I want to thank you, Diana, for all of your insights and the time that you took to walk me and the rest of the audience through this. It was fantastic. Thank you.
My pleasure. Thanks.
So with that, we are ready to take questions. Yeah. Thank you, everyone.
Great, thank you, Dan, and thank you, Diana. As a reminder to our participants, if you have a question, you can type it into the Q&A box in the control panel. The first question we have here, Dan, what is the turnaround time for testing? Can the results be returned before administration of systemic first-line treatment in metastatic non-small cell lung cancer and for neoadjuvant treatment in early stage non-small cell lung cancer?
Yeah, good question. So the turnaround time, this is, again, a tumor-informed approach. So the turnaround time for the first time point is approximately 3 weeks. That gives us the time to sequence the genomes, create the panel, and sequence the first plasma time point. All subsequent time points are about 7 to 10 days turnaround time.
Got it. The next one we have here, how many post-operative time points are needed to confidently define molecular clearance or molecular relapse?
I'll take this one, if that's okay. I think this question is a little bit more complex than just the number of tests. Again, tied to what I said during the presentation, it's very important to understand what the question is so that we can better utilize the best resources to answer it. So if you want to be assured of your answer, obviously you would want more than one. And again, it depends on the question, because if you're looking at moving at speed, then would you risk just testing one time point post-operatively, for example, in order to escalate patients with a ctDNA positive outcome? So the answer is not as simple, but I think that there is some room for us to better understand there by ensuring that we have the right question to answer.
What do you think are the next use cases to gain broader adoption for ctDNA in lung cancer versus breast cancer? Do you think they're different?
Yes. I wish I had a magic ball, right? To just make sure that I can see how the future looks like. But I think that there is a lot of momentum with respect to the data of the body of evidence building for the use of MRD and for escalation. I know that, for example, again, not talking about lung and not talking about breast, but in bladder cancer, we had IMvigor011 come out with the use of post-operative ctDNA status to identify those patients that had MRD positive and would go into one treatment versus the MRD negative. So I think that's where a lot of the data is pointing. And yes, lung and breast and in this bladder, CRC, they're all different. They have their nuances. So it's important to understand what the biology of the disease is to better understand how to best answer those questions with ctDNA. I don't know, Dan, whether you want to add anything to that.
No, I think you answered it well.
What percent of detections would you want to see after surgery to use a ctDNA assay for adjuvant therapy decisions?
I'm not sure I understand the question. Maybe I can rephrase it and answer that. So the percentage of detections, in the case of a post-surgical setting, which I think is the question here, I would feel comfortable with complete clearance. So not necessarily looking at anything less than a 50%. But then again, that depends on what the next step would be. So if you wanted to utilize it for adjuvant therapy decisions, I would want to make sure that there is a big difference, basically. I'm sorry, the question was just a little bit vague.
Yeah, and if you want to, the viewer who wrote that in, you can feel free to write in a clarification if you'd like. Are the regulatory requirements for validating MRD as an intermediate endpoint the same as those of establishing MRD as a surrogate endpoint?
Yeah, so what we saw in multiple myeloma is that you have to undergo the same process, the same meta-analysis. Obviously, you're aiming for the utilization or the validation of your biomarker as a validated surrogate endpoint. In the case of multiple myeloma, they looked at it and they determined that the evidence was there for it to be a prognostic biomarker, and thus the evidence pointed towards that, not so much as to the validated surrogate endpoint, but rather the intermediate endpoint. So it's not like you go and you say to the FDA, I want to be validated only for the intermediate endpoint, or I want to be validated only for the validated surrogate endpoint, but rather this is the data, this is what it shows, and this is how we're going to utilize it in the pathway, the regulatory pathway that we're going to make it available to.
Mm-hmm. How much reduction of ctDNA levels compared to baseline would be clinically meaningful, either during adjuvant treatment or post-curative intent therapy? For instance, is a 30% reduction meaningful, or are you only looking for clearance?
Yeah. Dan, I want to take this one because I also have an opinion about it.
I'll give my opinion, and you can give yours. So what we see, I mean, across a lot of different ctDNA studies, particularly in the late stage, that the overall reduction in ctDNA, like whatever that threshold is, whether it's 30%, 40%, 50%, is variable from one cancer type and one setting to another. For example, there was a melanoma study that Personalis was part of with Klaus Mantel and UKE, where the highest correlation to outcome was, I think, a 50% reduction, if I'm not mistaken. Maybe it's 30%. And then there was another study that we did with VHIO in Spain, where the reduction, the threshold was different. It was higher, like I think the first one was 30% and the second one was 50%. So my take on this is that there's never going to be one threshold. It's going to be dependent on the cancer type and the target population. That can be variable from one to the other. What do you think, Diana?
Yeah, I think that we have precedence. So what I was going to say was that as we are utilizing these ultra-sensitive tests, 30% drop can mean a lot. Whereas in the past, when we had seen an initial 30% drop with less sensitive tests, because you are seeing such a high baseline level, a 30% drop is not so much of a significant drop. But if your baseline level is really low, a 30% drop could be significant. We need the clinical utility studies to be able to answer that question.
Well, that brings us to the top of the hour. Unfortunately, that's all the time we have. We do have a few more questions here. We'll be able to provide those to the sponsor to hopefully follow up. Thank you to Diana Vega and Dan Norton and our sponsor, Personalis. If we didn't get to your question, we'll pass those along. Please take a moment to complete the survey that will appear after you log out. The first 100 live attendees to log in today will receive a $35 DoorDash gift card, which will be sent tomorrow afternoon to the email address used during registration. If you missed any part of the session or if you'd like to revisit it, an on-demand version will be emailed to all attendees. Thank you so much for joining us.